For those in the audience who aren't as familiar with IMPC, I think it's important to sort of put it in a historical perspective of these sort of milestones that have occurred over time that have enabled a project such as the IMPC to launch. And this really starts out with the generation of the first knockout mouse, back in 1989, and then with that, this ESL technology, people being very comfortable working with manipulating these cell lines and thinking about functional genomics. How do we screen the whole genome for unveiling gene activity? And ENU was a major powerhouse for antigen genome, and a lot of the pioneers of these techniques, were the architects of this knockout this project, and building upon the backbone of the mouse genome sequencing being available. There was this international effort to generate an ESL resource that represented the complete protein coding catalog that, as we knew it, and that's known as the International Mouse Knockout Consortium, IKMC. And so once this resource became available, UMODIC, as a European effort, really kind of put into place these large-scale phenotyping screens, so that showing the feasibility of being able to systematically put these mouse through a pipeline. And in the U.S., an effort known as the Knockout Mouse Project, in phenotyping program, went on to also mirror this, and then everything kind of got put under the umbrella of this International Mouse Phenotyping Consortium. And now we're in this moving past this timeline into a phase 3 of COMP2. And as these technologies have changed over time, we have quickly adopted them, and you'll see this reflected in the types of alleles that are present within our catalog. So as ESLs, were a major power horse, house for us, the advent of CRISPR-Cas9 being able to readily be able to adopt this into the laboratory, we quickly jumped on that and have been utilizing that as the primary driver for generating our models. So we think about the allele collection that's used for phenotyping. We have two major classes. We have the ESL resource and the Cas9 generated resource. And it's very important to know that these are all done on isogenic strains. So this is a C57 Black 6N substrain. And importantly, all of the mouse lines have extensive genotyping quality control before going to phenotyping. And then, what this does is it allows the people who are going to take up these mice to be ensured that they should be able to reproducibly repeat these phenotypes that we observe. So the ESL resource, some of the highlights there is that, given the changes in technologies and ways different groups have done this as an international effort, there are some different targeting strategies that I'll outline. But importantly, they include a Laxi reporter, which allows you to monitor the activity of that gene as endogenous locus, and then conditionality. So this enables one to use Cre-Lox technology to delete this gene in a specific time and place. And this resource has not been exhausted, so clones are still available to purchase. This collection of alleles is over 5,000 and was recently published a couple of years ago. And as you can see here, if we look at the conditional alleles, what IMPC has been able to bring to the table is almost 3,000 more conditional alleles to what had been already previously done by the, just a community effort resource. And then along with these alleles, we brought these reporters. So now there's over 4,000 new reporter strains that are out there, and some of that activity has been cataloged and available to browse in the IMPC portal. More recently, we've transitioned to using Cas9 as the primary driver. And what this allows us to do is directly engineer mouse zygotes. So it eliminates some breeding steps that were needed before, and we're directly modifying the Black 6N genome using a CRISPR-Cas9 technology and electroporation. And this opens up some different gene models, right? So if we get towards genes that have more complex structures, we have more flexibility in how we approach them. And it, more importantly, allows for a higher throughput at a lower cost. So like every effort, there are constraints based upon funding, and this allows us to cover more ground. One of the barriers I think people come across, that because there's a nomenclature that's unique to these gene these alleles that we've generated. And here I just like to provide a brief overview of some of the things that are coded in this nomenclature. So this is a standard design here for an ESL-based targeting construct where a critical exon is flanked by these LOXP sites, and it has this reporter, this LAC-Z, NEO, or beta-GEO, surrounded by these FRT sites. So this is a knockout first allele, in that splicing would splice into the selection cassette and then terminate transcription, and you wouldn't get translation of these downstream exons. There is some leakiness potential here, so for phenotyping, we cross these lines to a CRE expressor, a germline CRE, often SOX2 CRE here at JAX. And this 1A becomes a 1B. And you can see, for an example, this gene RAB34, there's information encoded in this superscript. So whenever you're looking at these alleles, there's a lot of information that you can gain just by looking at these superscripts. So that, you know, this is the TM1B in the collection of ESL resource that came from as part of the UCOM tools set. And if to make this ESL resource conditional, one could cross this TM1A with a flip expressing line, which would remove this selection cassette and reporter and leave the exon with LOXP sites flanking it. And then one could convert this TM1C to a TM1D to when it's crossed to a tissue-specific CRE. And this TM means that's a targeting mutation, meaning that it was intentional. You also see these TM1Es, which means that they lost this distal LOXP site upon recombination. So they've lost the conditional potential, but they're still targeted null potential. So these are some of the things you'll see in the ESL resource: A, B, C, A's and B's, and occasionally E's. C's and D's would be more investigator-driven lines of research. There's a couple more that are in the catalog, those which are just straight deletions of a critical exon. So this is a targeted deletion without the potential for extensive modification through the ABC nomenclature. And this TM1A, TM1 becomes a TM1.1 when crossed to CRE, or TM1.2 when crossed to flip. So this dot number indicates, a modification on top of the parental allele. You can see it here. This was another set of alleles that are present in the catalog, and these were generated by the VelociGene technology from Regeneron, where they took a very these were done prior to having whole genome sequencing and not knowing what the intervening sequence was. So they just went from start to stop, and they removed all the coding sequence between it. So this is a large deletion using bacterial vectors to promote the homologous recombination, and it has a selection cassette. So when you cross this line to CRE to remove that selection cassette, that TM1 becomes a TM1.1. And these are all indicated by this VLCG, that these are part of the VelociGene catalog. With the CRISPR lines, we have a little bit more flexibility, I mentioned, and as things evolved, we tried different strategies. So you'll see that there are different types of alleles. Initially, we used these single guide cuts, which are would cause a small indel. So 2 out of 3 would be out of frame. We can also use pair guides to create deletions, whether these deletions are within a critical domain of a critical exon, or they are removing the critical exon itself or the whole gene, mirroring the LOXP alleles. The primary focus now is doing these paired guide strategies and creating deletions with going up to 10 kb. And we're very careful in how we select these guys. So these are all selected to minimize the off-target risk. So we impose some hard filtering on that. We don't allow guys with 3 with 1 or 2 mismatches. We allow up to 3 mismatches, but nothing less than that. So we're very strict on how we select these. And then we also perform Sanger sequencing to get a precise identification of the nature of the deletion. So over time, you can see that we've done a lot of production as a consortium. Going back to the very early ESL resources quickly ramped up and has reached its plateau as Cas9 came to be the major player, and collectively now we're over 10,000 genes. So this represents more than half of the protein coding fraction of the genome that there's a IMPC allele for. And we try very hard not to duplicate efforts. So there are some genes that have both a Cas9 and an ESL allele, but it's very rare. And what we're moving towards, as a community, the ultimate goal, right, is to create a comprehensive catalog of mail and gene function. And I think this is both the IMPC as well as investigator-driven research. And if you look at the collections of alleles that there's a lot of ESL genes covered by ESL lines from community, and then the IMPC I mentioned we had another 5,000, and some were done by both. And we've quickly ramped up the number of Cas9 alleles. And we think about what is maybe the most human health relevant fraction of the mouse genome is those that are share human ortholog. So that brings the number down to about 17,000 protein coding genes. And collectively now we're, almost up to 14,000 genes that have been knocked out, leaving a little over 3,000 genes left over. And this is really where we're trying to put our emphasis, thinking about ways, how do we prioritize these genes, thinking about, we can use biological network-based evidence, we can use human disease, we can use constraint in the human genome. We always have some center-specific interests, disease models, and then, are these genes that we've attempted before, but then failed, or was there a biological reason why these were refractory to our approaches? So this things that we think about and going after this sort of understudied fraction of the genome. I think this is all kind of spelled out nicely right on the title page of the IMPC website, where the big bullet points are that all the data that you'll see in the database is from primary data generated by individual centers. So it's not aggregated from publications. We're really looking into these darker regions of the genome, things that we know very little about. I've shown you that we have very standardized production methods that we use to generate these knockouts. And we have very strict phenotyping protocols that are shared across this whole consortium, and mutants and wild types are always run in parallel. So the latest data release just came out at the beginning of May, and it has over 9,000 lines representing over 8,000 knockout genes, and over 100,000 phenotype calls. So this every data release, it's just really impressive to see these numbers just continuing to grow and grow. And so for the phenotyping, it's, as I mentioned, it's a very standardized pipeline, and the primary pipeline is what we call this early adult pipeline. And this covers a whole range of tests, things that look at behavior and gross morphology, strength, memory learning, and cardiovascular function, and hearing, and body composition. And then as these move to terminal procedures for necropsy, we collect blood and do clinical blood chemistry. And we also, there'll be a subset of data in the database that was done from a pilot aging study to see what impact, if you age out these knockout mice past a year, what do more phenotypes become evident. And almost 600 genes have gone through a late adult pipeline where the same battery of tests was repeated. And these data are available as a separate feature on the portal. In addition to these adult phenotypes, we also have a very extensive embryonic pipeline. So any mouse line that het by het mating fails to produce homozygous animals, we then put them through an embryonic pipeline to look at the impact on development, with the entry point at E9.5 as a first time point, and then moving through gestation. And here we assess the gross morphology of the embryo. We've formed some imaging, micro CT primarily, and then lines that are subviable, which is functionally defined as a het by het mating that produces less than half the expected Mendelian ratio, so less than 12.5% homes at wean. We also look at these through a pipeline where we're primarily using micro CT as a imaging modality. And here's just an whole image of an embryo, and then some of the LAC-Z data. So all those lines that were had the LAC-Z potential, and then we had embryonic pipeline data. We did the LAC-Z staining, so we had expressed some impression of where it was expressed in the embryo. This is a really widespread effort. It's a global effort. So I'm at the Jackson Laboratory and just representing one small piece of this larger puzzle. And it's really great to have all these colleagues spread throughout the globe, and we meet monthly to make sure that everything is on track, and we're broken into these different subgroups. Where production is just one aspect of it, but there's other groups that focus on specific phenotype domains and data analysis. So that is a pleasure to give this broad overview of animal production, and you can open up the floor to any questions.